got history of genetics. Genetics is a science of heredity and the mechanisms by which traits are passed from parents to offspring. So it's the study of the entire process of inheritance of traits. Heredity is the actual transmission of characteristics from the parents to the offspring. So this is like your parents passing on the traits to you. Genetics is the study of all of that. Okay, so we've got Gregor Mendel. He is the most famous scientist when it comes to studying genetics. He was in a monastery and worked in the garden, and he did a lot of studying. He studied science and math, and with that, researched heredity. Pea plants are the most used plant that he had for studying the traits because they were very simple to follow, but they had lots of characteristics that he could consider. So there were 7 characteristics that he studied: the shape of the seeds, the color of the pods, the shape of the pods, how big the plants got, the position of the flowers, and the flower color. So you can see that he identified dominant and recessive traits. We'll get more into that as we go. So a characteristic is a heritable feature that, such as the color, the height. Each characteristic had 2 differences. There wasn't an in-between, so either it was tall or it was short. A trait is genetically determined difference or variant of a characteristic. So tall or short plant, or for humans, a widow's peak or a straight hairline, those would be considered traits. It's what you physically see that can vary from one person to another. All right, so Mendel's experiments, he collected the seeds from the pea plants and recorded the traits. He observed patterns from season to season, compared them to the parent plants that they came from, and then eventually he got to the point where he started controlling the pollination so that he could see what happened if you cross-pollinated different colors or different seed types or different heights and see what happened. So self-pollination occurs when the pollen is transferred from the same plant. That would be an asexual reproduction where the offspring should ideally be the exact same as the parent was. Cross-pollination occurs between 2 plants, so there'd be essentially a mother and a father. Pea plants can self-pollinate but can be controlled by removing the anthers. So if you look here, if you remove the anthers, as was done here, it then prevents it from being able to fertilize itself. So what Mendel did is, in order to do the cross-pollination to see what would happen if he mixed 2 species with different traits or the same traits, he took the pollen from this plant and transferred it over to this plant in order to get them to reproduce a new offspring. So, for his experiments, he created and crossed true-bred plants through the self-pollination until he was sure that all of them were purebred or homozygous or true-bred. You probably won't hear the term true-bred very frequently. We'll be focusing more on the purebred and the homozygous. Remember, when we talked in previous units, the prefix homo means the same. So it's just implying that it's going to be the same alleles, which we'll talk about later. So essentially the same genetic information from both parents. He called the true-bred parents the P generation. So he was starting with purebred. If you're familiar with specific animals, if they are a purebred, there's papers you can get to prove that they're purebred, which tends to make them more expensive. The offspring of the P generation are called the first filial generation or the F1 generation. We'll mainly just be knowing that as the F1 generation. So we've got the P generation, F1 generation, and then it would be F2 generation for as long as the plants are studied. So here we have our P generation, the parents, the first filial, and the second filial generations. All right, so his results, you can see flower color. He could determine the dominant recessive forms. If you mix the purple with the white, the dominant would have 705, the recessive 224. Notice the ratio was about 3 to 1. And when he did the same thing with the dominant and recessive here, he had about 3 to 1. And all of these traits, his ratio ended up about 3 to 1 for every single one. So in one of the experiments, he crossed a purebred green seed and a purebred yellow seed. All of the F1 offspring were yellow, but then when he got to the second generation was when he was seeing the consistency of the 3 to 1, where 3 were the dominant and 1 was the recessive. But it took 2 generations in order to see that. And hopefully we'll come back to that when we talk about the Punnett square so that you can see how that works out. Mendel determined that alleles controlled the characteristics in the plants. We'll get into alleles later. We'll define it a little more when we talk about that in the next lesson.